Can you hear me okay? Anna Laurence?
Yes, hello. You hear
Hi.
me?
Yes, I can.
Good.
Thank you for joining me.
It's a real pleasure for me.
We had a little bit of technical problems with our Google Hangout, but I think we're good to go now. So,
Good.
are you in Paris?
No, I'm in the south of France, in Toulouse, in fact.
Toulouse. Oh, very
Yes,
good. Very good. Well, thank you for your interest in medical neuroscience and for, I'm sure, all the hard work you've done so far in the first 2 weeks for us.
sure. But I want to thank you too, because it's really wonderfully interesting.
Good. I'm glad you think so. And we're just getting started. I think it's going to get even better over the next several weeks.
Yes.
Is there any particular question on your mind?
Well, I have many, but maybe one in particular about neurons that fire together, wire together, the sentence from your colleague and friend. I must say, what I do not understand, among many things, of course, is there always a percentage of neurons within each individual that sometimes don't fire together and thus do not wire together? And is there a way, well, is it the case or not? And is there a way to improve that or to monitor it by exercising or whatever? Well, whatever you can tell me about this is of interest for me.
Sure, thank you. Those are excellent questions. So in order for neurons to truly wire together, they must have some connection or at least be in close proximity such that they can establish an anatomical connection. So that's one requirement. If you're talking about neurons that have no axon near a dendrite, then there's not potential for wiring together. But assuming that there is a pioneering axon that's beginning to establish at least an exploratory contact with the postsynaptic neuron, now that aphorism about firing together and wiring together becomes possible. So essentially, most neurons are not interconnected, of course. So yes, it is possible to have neurons that are not anywhere near each other and have no functional relationship to each other, and therefore no compulsion to wire together. But among those neurons that are in close contact, there is much exploration that happens between a dendrite and an axon. And there are connections that form and break and form and break all of the time. In fact, perhaps most connections, particularly in, let's say, the first 1 to 2 decades of life, are of these exploratory kinds of synapses. And if there is coordinated firing across pre- and postsynaptic elements, then that's where the wiring together can happen.
Yes.
As neurons begin to fire more and more. So how can we promote that? Well, I think we should consider Hebb's postulate, which is captured by this saying of wiring together because of firing together. We should also think about spike timing-dependent plasticity, which we can think of as basically connections that are already becoming well-wired get even stronger, and those that are not well-wired tend to be eliminated. So how can we strengthen the moderate connections, get them more functional, more adaptive? I think we don't know a great answer to this question, but the more consistently we can repeat patterns of neural activation, the more we can promote this adaptive plasticity that leads to wiring together. So what does that mean in real life? I think, for example, if you're learning a skill, perhaps playing a musical instrument or perhaps learning a new dance, the more one can repeat the same pattern of sensory motor experience, then we have the ability to increase the firing together that leads to wiring together. And this is a real challenge because most of us in our real world activities, we don't have the patience or the time or the commitment to truly repeat and practice over and over and over again. Thankfully, many people can, and so that's where expertise comes from. But for many of us, it just does not, it's very difficult to devote sufficient time and energy to repeating the experience sufficiently to really optimize neuroplasticity that leads to the acquisition of skill and increase human performance, or in the context of rehabilitation from injury, successful rehabilitation from injury.
Okay.
So it's a complicated question, really, and I'm sorry I can't give you a more satisfying answer, but I think if we satisfy the conditions of Hebb's postulate, then we are more likely to see efficient adaptive plasticity, and with it the acquisition of skill.
Okay, so it means that if it's, and then I stop, it's the uncoordination or the lack of coordination can be corrected, in fact, by exercise and exercise and exercise.
I think so, and I think the quality of the experience is really important. We will actually talk more about this when we talk about the development of the brain and the plasticity that happens in early life. That comes up in Unit 5 of the course, but for now I'll just say that, to use Hebb's terms, the coordination of pre- and postsynaptic activity comes when we reduce the variance. That is, when we increase the precision of our experience, be it a motor act or some kind of sensory experience. So if we could really practice the same skill over and over and over again and become increasingly precise, then in our nervous system we are very likely reinforcing the connections that are instantiating that skill, and pruning away the connections that are exploring the variance or the variation around that skill.
Okay, thank you very much.
Okay.
So I have to disconnect now.
Well, you are the only person in the Hangout at the moment, so.
No, really.
One of the things that happened since our last Hangout, Google has totally redone its interface. So we're trying to understand how to make this work from our end. So I apologize to you and anybody else who may be watching right now that we don't have this worked out. But since you joined us, we know that something must have worked.
Yes.
So
obviously. Okay, so maybe I can ask you something else, or
yes,
what?
Please do. Yeah.
Or maybe you have Q&A on the screen, or no? No, it's about the memory, the loss of memory of our very, very early childhood. In fact, I don't know if you are going to develop that in the Unit 6. Well, I've been through Unit 6. I will not really see it, but I have read many different theories about we produce too many neurons, so we are not able to manage them, and then they all die, and well, so I don't know what happens. So maybe if you could elaborate on this just a little. I see somebody else.
Yes, some more people are joining us. Welcome. So you're basically asking, why do we fail to retain our early life memories? Is that what you're asking?
We fail to, yes, exactly.
Yeah.
Or maybe we don't. And
yeah. Well, I think most of us begin to recall early life sometime around 3 years of age. Maybe some a little before then, but usually around 3 years of age seems to be the common period where our memories become more permanent. And I think that simply reflects the ongoing construction of circuitry in the brain. So in those first couple of years of life, and really throughout much of the first decade, the brain is an incredibly dynamic place. There are connections being formed and broken all of the time. And we can think of the formation of new connections as a way of making our memories more durable. Maybe not permanent, but at least more enduring. And so if the brain is constantly probing and pioneering new connections, some of which stick, some of which don't, then we have limited capacity to store information. So I think part of the cost of building a large brain that's richly interconnected in early life means that we have limited capacity to store information for the very long term. Now, part of it also is that not all brain areas are developing at the same rate. So those parts of the brain that are elaborating cognition and eventually storing memories are a bit later to really fully mature. So the rate of growth is just different, and the rate of circuit construction is probably different. So I think that most of our early memories are more of a sensory nature, and then we begin to elaborate over time. But the other fascinating issue about memory is that we change our memory as we go through the process of recall. So every time we remember something, even from our distant childhood, we have the capacity to alter that memory and its structure because it's subject to ongoing plasticity. And so the psychologists have studied this and have shown how people's memories can actually evolve over time, even though the person may still have a very profound conviction that, yes, this is exactly the way it happened, but really some of those details can change. I'm sure I'm guilty of that myself as I tell stories from my own childhood.
Thank you very much.
Okay, so thank you for joining me and Laurence.
Okay. So I disconnect now, okay?
Yes, you can continue to watch, but you can disconnect from the Hangout.
Okay, I don't know how to do that, but I managed. Thank you very much.
Okay, you're welcome. So let's go to Rafa. Rafa, welcome.
Hey, actually it's Rafael.
Oh, Rafael, okay.
So I'm from Brazil, and I'm a medical student here, and I really want to thank you for taking your time for doing this course for all of us, your students from all around the world. And actually, as a future physician, I think it's important to understand some things, and I actually want to know what's coma and why some people get in coma and can get out and others can't.
Yeah, well, thank you for your participation in the course. I was just looking this morning, and I realized that Brazil is the #2 nation in medical neuroscience right now. So thanks for your participation. Maybe you can encourage some other friends to join us either now or in the future. But thank you for your question about coma. So coma is a state of brain and body where there is life, there is the necessary functions, particularly of the brain stem, that are sustaining the physiology of the body, but there is very limited activation of the cortex. So the cerebral cortex is largely in a state that is incapable of supporting conscious thought. Now, we talk about coma as if it's very clear and it's really obvious when a person's in a coma or not, but that's really not the case. There very well may be a kind of mental life that is present in someone that is in a minimally conscious state. So sometimes we struggle to differentiate a coma from a minimally conscious state. And there have been some really interesting studies that have been done on some individuals over the years where people have been thought to be in deep coma, maybe even near brain death. And it's been discovered that they actually can modulate brain activity in a volitional way in communication. So I'll just tell you quickly about one study where an individual was considered to be in a minimally conscious state, but this person was asked questions while in an fMRI machine. So the person was a tennis player, okay? So the person was asked, think about playing tennis. Well, when the person was asked that, the premotor cortex became activated. And then they asked the person, think about walking through your home. And when the person was thinking about walking through their home, their parietal cortex was activated. And now the investigators realized they could communicate with this person in a minimally conscious state. So they would ask questions like, are you hungry? If you're hungry, think about playing tennis. And they could see that the motor cortex were activated. And then they would say, okay, if you're hungry, think about walking through your home. So now they saw a parietal cortex. So they know they could confirm that the person really knew what they were asking. And then in this particular case, they eventually asked the person, do you want to live? If you want to live, think about playing tennis. Well, now the premotor cortex is activated. So here's a person that our medical system might have thought is in a deep coma, possibly in such severe cognitive impairment that we might consider this person to be brain dead, when in truth this person was very much alive and very much engaged in mental processes. So how does this happen? Well, this can happen with either destruction of the cortex itself, or at least severe damage to the cortex, or it can happen with damage to the systems in the brainstem that alter the state of function of the forebrain. So there are nuclei that you're probably learning about in the core of the brainstem that are part of what's sometimes called the reticular activating system. And if these key nuclei are damaged, among them are cholinergic nuclei, noradrenergic nuclei, then the brain state of consistent activation that we expect when we're awake can be compromised and we can slip into coma.
And why do you think some people can get out of coma and others can't?
Yeah, well, it often depends upon the extent of injury to the cerebral cortex. So one could be in a coma where the cerebral cortex is essentially intact and healthy, and the problem's in the brainstem. So if those cells in the brainstem can somehow recover their function, then they may continue to activate the cortex. And those people that have had damage to the cortex have less opportunity to recover. But again, this is a continuum. No two people are going to be the same in the extent of their injury. And I think what we've learned in neuroscience and in neurology is that we should never underestimate the power of the brain to recover and to undergo plastic change. So finding ways to somehow promote plasticity is going to be very important, even for people in minimally conscious states.
Thank you very much for your consideration.
You're welcome. Thank you for your participation in medical neuroscience. Okay, I think we'll go to Amin. Am I saying your name correctly?
Hello.
Hi, where are you?
My name is Amin.
Amin, welcome.
Hello, I'm from Iran, and let me first thank you for all of your efforts in instructing this amazing course. And I have a question. And there is a hypothesis which says that through the evolution, the human brain is changed in such a way that it is performing tasks efficiently in our living world. Based on this hypothesis, I have two questions. First, what do you think about the efficiency of brain in performing tasks? I mean, there are cellular spontaneous activities even when we are sleeping. And my second question is that are there inputs in this world that we are not able to sense? I mean, do you think that we have all of the sensory inputs needed to receive every single event in this world?
Thank you, Amin, for your questions. Thank you most of all for your participation. It's really a privilege to have you in the course. Let me say that in response to your first question, that actually, I think I'm going to ask you to rephrase your question because I just want to make sure I understood you correctly. So could you say your first question one more time?
I was thinking about the efficiency of the brain in performing tasks. Is the brain efficient through the evolution?
Yeah. So the concept of efficiency is always a challenge because on the one hand, you might say, well, the brain is very inefficient. It requires about 20% of the cardiac output, yet it only accounts for about 2% of our body weight. And the brain itself has about 85 billion neurons and probably about that many glial cells. So that seems to be an overly redundant organ that is requiring way too much cardiac output to support. So from that standpoint, one might think that it's inefficient, but yet it's the organ that has evolved. So based on principles of evolution, we might say, well, there must be a benefit that has allowed for a certain degree of inefficiency. So I think that what we've learned about the brain over the years is that it is highly adaptive, and with its adaptation gives us the advantage to accommodate to changes in our environment, changes in our world, changes that we ourselves impose upon it. And some of that provides some advantage in adapting to the life experiences that we face. So I think we have this amazing repertoire built into the brain in a highly dynamic and highly flexible way. So I think there has to be some kind of inefficiency that will give us the capacity to respond to the challenges we face in life. So I think if our efficiency was less than it is, we probably would be less successful as a species and probably less successful individually. But surely this has come at a cost. You know, we have these large brains, these large heads. We have, you know, the whole process of being born is a major problem because of the size of our brain, right? And so the evolution of the human gestation period seems to be matched for the size of our head, the size of the birth canal, and yet as we enter life, we encounter these dynamic, complex situations, some of which are environmental, some of which are social, that demand this capacity to adapt and to make predictive models, which is something the brain is extraordinarily good at. So I think the inefficiency really serves us well at key points in time. Maybe it doesn't always serve us well, but I think our ability to solve problems, to engage in social relations, has a lot to do with understanding the brain that we have. So, very good question. I'm afraid that I've kind of forgotten your second question, so remind me of that, please.
The second question was that do you think that we have all the sensory inputs
Yes.
needed to repeat every single event in this sport, or are there events in this sport that we cannot sense?
Sure. Well, we know that our sensory channels are tuned to a very narrow band of the energy that is present in the environment and the energy that we, our body, create. So I think the answer is that we are not aware of all of the energy that surrounds us. So that's is just a fundamental fact of the nervous system that we have. So for example, when we consider light, even though we respond to about 10 or 12 log units of radiance in the environment, there's still the infrared, and there's still the ultraviolet, and the rest of the electromagnetic radiation that we cannot sense. Some creatures do a better job of sensing certain bands of that energy, but we are limited. And the same is true of audition. The same would be true in somatic sensation, and even the chemical senses. There is limited bandwidth of sensitivity. So I think it's just a profound fact of evolution that we are tuned to survive and perhaps even thrive in the environment in which we live, but that has come with a kind of canalization of our ability to respond to the energy that's all around us. Now, our brain still has the capacity to do more. We know this from some new experiments where scientists have added additional senses. So, for example, I have a colleague here at Duke who has created a rat that has an infrared detector mounted on his head, and the output of the infrared detector is stimulating the somatic sensory cortex. And so these rats that have been so modified are learning very rapidly to use infrared energy, which they cannot see, to guide their behavior. So there's a case where an animal has been given an extra sense, and yet its brain has readily enfolded it. I think this also happens in the motor domain. So, for example, as people that operate tools, or people that play sports, or people that operate heavy machines, they have this amazing capacity to begin to expand their brain's representation of themselves to include these various devices, be they mechanical or instrumental. So I think the brain remains highly adaptable, and our experiences as we gain expertise manipulating our world become enfolded in the way the brain represents itself.
Thank you very much. Thank you for your consideration, and I hope to join your New Year run someday.
Okay. Well, I'd love to have you. I'm going to show you all something. So I had a Neuro run this weekend, and unfortunately I broke my foot on my run. So I won't be running for the next 2 weeks. But I look forward to getting back out there.
Maybe this time you can watch me running.
Okay. Thank you, Amin, for joining us. Okay, let's go to Joanna. I think you've been waiting for a few minutes. Hi, can you hear me?
Our mic's on.
I think
It's on now.
Okay, thank you.
Yes.
Go ahead, Joanna. Do you have a question?
Oh, yes, I do. Can you hear me?
Yes, I can.
Oh, I'm really sorry. My connection is not great today, so I'm really sorry about that. So first of all, thank you very much for the opportunity. This is great that you are organizing this Hangout. The course, it's fantastic, a lot of resources. The topics are great, so thank you very much for that.
You're welcome.
And my question is about everything that is connected with communication and particularly with speech. So if we have a normal brain, if I can say normal, if I can use the word normal. So if you have a normal brain that the anatomy looks perfect and the physiological part too, how it is possible that a person with this profile doesn't have the communication working? So I'm talking about in particularly the verbal communication. So how is that possible?
Very interesting question. I think there are many possibilities. One might have a functioning network in the lateral temporal cortex for understanding language, but perhaps the connections to the part of the frontal lobe that makes language are somehow dysfunctional, or perhaps the wiring just is not as typical as we might expect it to be. That's a possibility, essentially, that there is a connectional issue with the way the brain developed. There may be a problem that's more isolated to either the receptive side of the language system or the productive side of the language system. There may also be issues related to how our prefrontal cortex, where our executive control centers reside, interact with our language system. So we may have thoughts. They may be quite rich thoughts that give us a rich inner voice, but if those systems of the frontal lobe don't connect well to the language centers, then maybe those inner thoughts don't become outer thoughts, voiced through our vocal motor apparatus. So I think what we're coming to discover is that there is a rich continuum in the human species with respect to how our brains are wired to connect our thoughts with our language centers. And probably no two people are identical in this way. There is a growing interest in this very question, particularly among those individuals that are studying autism spectrum disorders, where verbal fluency can be an issue. We also see this among individuals older in life that are beginning to experience cognitive decline. Often there is a loss of verbal fluency there as well. So what we need to do a better job is understanding how the inner voice is connected with our capacity to make that voice audible and then to engage in social relation with others so that our language experience can be productive. And I think what we're going to discover is that there's just a rich variety of architectures that enable different proficiencies for different individuals. So those are just some early thoughts. We'll talk a little bit more about language function in Unit 6 of Medical Neuroscience, and surely there will be much more to say in the years to come as we understand more about this part of the brain. So Joanna, I think I've lost you, but hopefully you're still listening. Let's go now to Marina. Are you there?
Yes, sir. How are you? Can you hear me well?
I can. How are you?
I'm doing well, thank you. So first of all, I would like to say that I've taken about 10 courses online now, and this has been exceeded all of my expectations, and all of the courses that I've taken were excellent, but this was just amazing, and it was one of my favorites just because of the topic. I became interested in the brain when I was about 15 and haven't stopped loving it since, and this was the first serious class that I have taken for neuroscience, and I'm just really excited. So thank you so much. You are an amazing educator. So, and added to that, we have the best in the course. We have the best teaching assistants.
We certainly do. Thank you so much. That's very kind of you to say all those things, Marina.
Thank you so much. Thank you for all that you do for us, and to your helpful staff. My question today would be, I have written several questions, but then I had another thought pop in. It's very interesting to me to hear something that you're passionate about. So you have mentioned in some of the videos about some of your past research, and I'm sure that most of my classmates would love to hear you speak about maybe maybe your most recent or something that you want to talk about, your own research. We would love to.
Okay, well, thank you, Marina.
Thank you.
My own research is a bit more specialized and what is well suited for the general topics of medical neuroscience, but I actually will say something about it in Unit 5 of medical neuroscience, because my research has largely been aimed at trying to understand the development of circuitry in the cerebral cortex, and in particular understanding how early life events, early life experience can shape those circuits. So one very conventional way to study these questions is to use the sensory systems and to use the visual system in particular. So that's what I've focused on in my research. I've done other things, but primarily the work that I guess I would say I'm most proud of has been focused on understanding the development and the evolution of the visual cortex. My most recent publications include a paper just a few months ago in the journal PLOS Computational Biology, which of course anyone's welcome to look at if they care to. But it has to do with understanding the development and evolution of cortical networks from a more theoretical and computational perspective. And I've collaborated with some wonderful colleagues, most of whom are in Germany, and they come from the world of theoretical physics. So it's been very exciting for me as an empirical neuroscientist to collaborate with theoretical physicists. And one of the fundamental discoveries we made a few years ago is that one can explain the organization of circuitry in the cerebral cortex with mathematical precision. And that's been very exciting because it suggests that there are some fundamental principles that help to organize the brain, and these principles are not encoded in the genome, and they are not necessarily fully determined by life experience. Rather, they self-organize because the brain is a dynamical network that has the necessary conditions for self-organization. And what we found in our studies of the visual cortex is a statistical pattern that characterizes the architecture of this space. And in fact, I have something on my wall which I can try to show you if I don't knock over all of my wires here. But I've got this image, which I think you can see the colored swirls there.
Mm-hmm.
We'll talk a little bit more about that in Unit 5 of the course, but that is a representation of the visual cortex, and all of those colors are part of a circuitry that we call the pinwheel. And there are actually many hundreds of pinwheels in that image over there. And what we did was we looked at the distribution of these pinwheels, and we found that they are distributed in a very statistically predictable way. In fact, the density of pinwheels in the brains of the animals that we studied is 3.14. Well, that should be a familiar number to you.
Hi.
Yes, so my colleagues who are the theoretical physicists have elaborated a computational model for understanding the self-organization of the cortex, and what they determine in their model is that the density of pinwheels is precisely pi. So the model shows that the repeating units are distributed at a density of pi, and what I found in the animals that we've worked on is a observed density of 3.14. So we think that's sort of a remarkable convergence of empirical observation with theory. So we're pretty excited about that and trying to understand what that means and what insight we can gather about how the brain evolved and how it develops in life. And so we think we've learned a few things in our recent studies of all of that. So again, that's a specialized topic. We won't really delve into that in medical neuroscience, but I appreciate your interest, and it's always fun to talk about one's own research.
Thank you so much. That is so interesting to me too, because I was a math major, and so the computational part sounds really cool.
Where are you,
Thank you so much for
Marina?
letting me join. I will yield to other.
Okay. Let me just ask, where are you? Oh, she's gone. Okay. So how about if we go to Linoy? Am I saying your name correctly?
Yes.
Hi. Welcome.
Hi. Can you hear me though?
I can.
Oh, okay, that's great. I'm really happy I got to join in because it's my first time doing this, and I'm completely grateful to the team and what you guys do. My question, I study cancer and cancer development on the DNA level, and recently I came to think that everything, since we want to achieve homeostasis, and homeostasis, it's it's quite on the physiological level. It requires all the wires work at once at a perfect speed and with a perfect timing. So my point with homeostasis is fluid imbalance. So when you have so much cerebrospinal fluid, so it causes certain diseases. So how do you look at water imbalance and homeostasis with relation to neuronal activity and the firing of the affecting the brain activity through water and volume imbalance?
Wonderful question. So one of the immediate thoughts that come to my mind is that when you think about water in nervous tissue, you think about ions and their concentrations. And so if you've been working through Unit 2 of Medical Neuroscience, you know how important it is to maintain a homeostatic level of some of the key ions that are involved in neural signaling, like sodium and potassium and calcium. So if one has some disturbance in the volume of extracellular space, one might alter the concentration of those ions. And of course, if you have some kind of disorder or disease or some kind of dietary insufficiency, one might also have an alteration in those ionic fluxes. So one immediate consideration is, how does the movement of fluid in and out of the brain influence the capacity to generate neural signals? But there are many other very exciting areas of active research about this. One of them has to do with some newly discovered lymphatic channels within the cranium that suggests that the movement of fluid through the cranium is very important for brain health and brain function. One idea is that this is modulated during sleep, and that during sleep there is a process that allows for extracellular fluids to drain out of the brain and to enter the cerebrospinal circulation. And this may be an important way of clearing the brain of potential toxic compounds that might impair, over the long haul, neuronal function. One of the really intriguing observations is that beta amyloid protein has been isolated from the fluids of an animal model that are cleared during sleep. So it's led to a very interesting hypothesis that perhaps part of our conditions that we associate with age, including Alzheimer's disease, may have something to do with fluid dynamics in the brain throughout life. So I think we're going to learn a lot more about this in years to come. But I do think it is an underappreciated aspect of brain function. Sometimes we like to focus on neurons and action potentials, and we fail to appreciate the brain as an organ. And as an organ, we should be thinking more about homeostasis with respect to blood flow, energy balance, and certainly fluid dynamics.
Wow, I would totally love to participate in that research and to kind of contribute. But I thank you for your time and your explanation.
Okay, well, thanks for joining us.
Thanks.
Okay, let's go to Anthony next. Hi, Anthony. Anthony, I can't hear you. Are you muted?
Can you hear me now?
Yes, I can hear you.
Okay, all
very
right.
clearly now. Thank you.
Sure. So I'm a licensed counselor, and I try to take more of a brain-based approach in working with people with mental health issues such as depression and anxiety. And one of my concerns is trying to increase the development of the prefrontal cortex for people with a lot of executive function disorders. And a lot of the recommendations that I have and things that I've learned over the time, I am just kind of second-guessing myself in a way about what is really effective, and in terms of intensity, frequency and duration of the exercises I'm asking them to do, such as meditation, exercise, cognitive training exercises that help promote the development of the prefrontal cortex so that they can see a resurgence of activity in that area, assuming that it was there before, or to get them to see that they have a higher order cognitive function available to them to help them in their everyday life. So, some of the things that I hear in just, I guess, colloquial or even semi-professional discussions about cognitive exercises to do usually say something like, for example, they might say for meditation, just do about 10 to 15 minutes a day. And I'm thinking that perhaps some of these these cognitive exercises are beneath the threshold at which a person is going to feel the effects of it. So if enough episodes of them trying meditation, they may come back to me like a week later and say, this whole meditation thing doesn't work. And I say, actually it does. It's just that I'm just wondering about frequency, intensity, and duration. So are these legitimate cognitive training exercises that help, and are we really, some of the recommendations are just too minimal to really start to see an effect?
Well, Anthony, first of all, thank you for your work. I think it's very important work. We need more people like you working to try to improve our mental health status. And it's just really, really critical, the work that you're doing. And I appreciate the nature of your questions, too, because I think that it's actually very likely that your insight is absolutely on the mark. I think that there is potential for all manner of cognitive behavioral therapy to really be transformative, and to do so not just on what we might say the cognitive level, but really the neurobiological level, really altering the function, maybe even the structure of key networks in the brain. So in order to get there, we really need to understand how do the principles of plasticity play out even in the prefrontal cortex, or even in the medial temporal lobe, or in other parts of the associational cortex that support cognition. So the principle of repetition, the principle of specificity and of precision is very important. So I think that finding ways to take those principles out of the sensory motor domain where we know the best and apply them to the cognitive domain is going to be a real challenge. But I think that most of us are unwilling to repeat and to dose our behaviors to the point that fully optimizes plasticity. So I'm just saying this as a matter of principle, not because I know the actual evidence base that would critique a particular intervention such as meditation, because I don't. But I would suggest that we don't take advantage of the need to repeat and the need to increase the duration and the intensity to a degree that would optimize plasticity. But I believe that potential is there. One other phenomenon that I would highlight, we will talk about this later on in the course in Unit 6, is that we know that among the many benefits of exercise and cognitive behavioral therapy and other kinds of therapeutic modalities for mental health intervention, including pharmaceuticals, is an increase in neurogenesis in the hippocampus. And it's very intriguing because that truly is where much of the benefit is being played out to increase mental health. So those things that can be done to optimize hippocampal neogenesis might very well be key to improving mental health. And among the interventions that have been studied in preclinical studies, the single most important factor seems to be exercise. So I find that to be very, very intriguing. I think still, in my estimation, the single best thing we can do for the brain is, in fact, exercise. And if we can somehow combine exercise with various kinds of therapeutic activities, including things like meditation or mindful movement and healthy social relations, I think some kind of multi-dimensional approach to wellness and health is going to be key. And obviously, you know, I think that mental health and physical health are really one and the same, and we just have different ways of thinking about them. They all relate back to the body-brain connection and how what's going on inside of our brain works itself out to the rest of our life and the rest of our bodies. So a long-winded answer to simply say that I think, Anthony, that your thoughts about what you're doing are right on the mark. They're neurobiologically sound, but what we lack is a robust evidence base to really begin to figure out, okay, how do we do this well to optimize plasticity, and can we actually measure something about the brain that provides us with a biometric outcome. But I think we'll get there. I think that our brain imaging methods and our ways of interrogating brain structure and function are improving all the time. So there's great hope, I think, to more firmly establish a biological basis for the kinds of therapeutic interventions that you're pursuing.
Thank you very much for that validation. I appreciate that.
Okay, you're welcome. Thank you, Anthony.
Lisa, what's next?
Let me all just show you. I'm working with a wonderful colleague here, Nick. So where are you, Nick? You're down there somewhere. There you are. So Nick is trying to help point me to who I should be calling on next.
Lisa.
So Lisa, go right ahead.
Hi.
Hello, where are you, Lisa?
I'm in Germany.
Oh, wonderful.
Yeah. Speak of the devil.
Uh-huh.
Yeah. So I was wondering if you could tell us maybe something about, because, okay, I've heard, I mean, I went to Berkeley, and now I'm studying medicine here in Germany.
Mm-hmm.
So it's kind of crazy. But I know that Einstein, for example, Marion Diamond did research and found that he had more glial cells than the average person. But then I also wonder, like, does that have anything maybe to do with self-discipline and studies like that? Because I also have read stuff from Stanford that talks about how you can improve your willpower and how exercising is also very helpful for willpower. Do you know much about that?
Well, you're asking some pretty deep questions, so yeah. I don't think we know quite the connection, but glial biology has really exploded in the last few years. So glial cells in the nervous system, they sit at the interface between immunological function and synaptic plasticity. So I think the fact that Einstein had more glial cells in his brain isn't so much the remarkable piece of the story, but what I think is remarkable is that it infers that perhaps he had an enriched capacity for plasticity, that the glial cells we now know are important players in reshaping the structure of connections in the brain, and depending upon the glial cell we're talking about, the glial cells such as microglia can help mediate interactions between the immune system that can have long-lasting impacts on brain structure and function. So putting all that together, I think our glial cells are really important aspects of the biology of the brain. Now, how willpower and other kinds of focus concentrations interact with those systems, I don't think anybody really knows yet. But I think to the degree that willpower is a manifestation of executive functioning in the brain, we can be sure that the glial cells are somehow involved in their partnerships with the neurons. So I guess my suggestion is that what's good for neurons is also good for glia, and whether there are particular activities that are especially good for glia has yet to really be determined. We know there are things that are bad for glia, okay, like immunological insults, infections, particularly in early life. Environmental pollution is bad for glia, and the impact of these insults, particularly in early life, even in prenatal life, can have long-lasting effects on the brain. This is some research that is being done by actually one of my colleagues here at Duke, Professor Stacy Bilbo. So it's becoming more and more clear how important these glial cells are. So I think, again, we should recognize that they are very important players in brain health and in plasticity. So if we want to truly understand how to optimize plasticity and improve our status with our physical and our mental health, then we should understand how these glial cells interact with synaptic formation and synaptic plasticity. And we're on our way to doing that. We just need more time, more research, more discovery.
Do you also think it may have something to do with diet? Because I know memory champions and stuff, they'll drink blueberry concentrate or something before a competition or things like that.
Yeah. Sure. Well, you know, there are lots of insults that our brain is exposed to all the time, oxidative stress. So, you know, blueberries have antioxidant properties, and it may be that we are reducing a bit of that cellular stress, and that might have a rather rapid impact on these dynamics that result in the formation of new synaptic connections that ultimately underlie memory. So, yeah, there may be a lot to that. And again, exercise is the single best thing we can do, potentially because it helps to reduce this oxidative stress and helps us, you know, reestablish a more healthy sort of homeostasis within our body and brain.
Great. Thank you.
Okay. Thanks for your question, Lisa. Good luck with your medical studies.
Thank you.
Said how, I don't know.
Okay. So let's go to How. Are you there? I think your microphone is muted if you are there. Okay, maybe not. So let's move on to Jacqueline. Jacqueline, are you there? I think you need to unmute your microphone, Jacqueline. Are you with us, Jacqueline? I see you there, but you're not talking. So maybe there's a technical issue. So let's move on to Virginia. Hi, Virginia.
Hi, can you hear me?
Very well. Thanks for joining us.
Well, nice to join you too. I wish I could join you in Duke Forest. I used to go to UNC, and that was one of my favorite hangouts. So
wonderful.
Maybe someday we'll get back there. Well, tonight I'm going to a presentation about neurofeedback, and I was curious what your thoughts are on the effectiveness. I guess they use a QEEG to get that data, and then through a series of exercises, I guess you're supposed to be able to strengthen those certain brain waves that are more conducive to executive functioning.
Sure. Well, I think there is a lot of potential in these kinds of biofeedback approaches, and I think it could be actually quite beneficial to gain some deeper insight into the state of mind and body. And this technique that you're referring to is one way to actually put some kind of instrumentation on this aspect of brain function. So I think that what we've shown is that people are remarkably adaptable and have shown really great potential to get this kind of feedback and then do something about it. Actually begin to entrain their thoughts and their mind and their body into a state that might be more consistent with whatever it is they're trying to achieve, be it more focused concentration or just a greater sense of inner peace, whatever that may be. So I do think there is potential to have this kind of adaptive technology be used to focus one's mind in a way to shift one's state of mind and body in a more productive way. So I look forward to what you discover. Maybe you'll be able to come back and inform the rest of the community what you've learned at this seminar.
All right, thank you.
Okay, good luck with all of that. All right, so who should we go to next, Nick?
Anybody's got any more questions? There are some that have been asked on the Q&A.
Okay, so we are nearing. We had agreed to do this for about an hour. We're just about there, but let's see if Nick has any questions that came in through our Q&A function.
So this one is for Magali Barrera.
Says, I would like to know what is the relation between culture and cognition, and if there is changes evidence in the brain.
Okay, great question. So a question about cognition and culture. So I think just about any dimension of human experience is very likely to have an impact on brain structure and function. Nick, we're getting a little feedback. I don't know if that's from the mic there. Okay, so the evidence to support this really comes from multiple dimensions of cognitive science. One can look, for example, in the dimension of language, which can be a defining feature of culture, and appreciate how the organization of language centers in the brain might be defined somewhat differently depending upon one's culture. One can look at the way our attentional mechanisms are allocated and find differences in the area of culture. One can look at, for example, some of what we were just discussing with Virginia, one's ability to regulate one's own brain waves and see that certain cultural contexts are particularly skilled in those kinds of approaches to mind-body regulation than others. So I think we'll see evidence from a variety of dimensions of mind and culture that intersect brain structure and function in really interesting ways. And I think much of this is adaptable, much of it is teachable. And so I think we have the ability to alter some of these dynamics as a result of how we experience the world and how we, through our intentionality, come to engage these various dimensions of brain function. So I think there's really a lot to be done there, and we're beginning to gain some pretty key insight, because actually this is a very important question, right? This has a lot of real-world ramifications. It has implications for how we view societies other than our own, how we experience implicit bias, or how we recognize that in ourselves or others. And I think really there are many social consequences that will come from a deeper understanding of culture and cognition as it affects brain structure and function. So thanks for that question. Let's see if there's something else. Nick, another question from the text world?
Is it possible that change in plasticity can lead to neuronal death?
Very good question. Certainly, synaptic plasticity can lead to a loss of connections. Can it actually lead to the death of neurons? The one context in which the answer very clearly is yes is in the developing nervous system. So we know that there are connections, for example, between motor neurons and muscle fibers, or between preganglionic autonomic neurons and postganglionic neurons, where there's a kind of competition. And there's a kind of competition that seems to be aimed at a trophic molecule, that is a substance that is secreted in a limited quantity by target tissue that's necessary to nourish and promote the survival of the presynaptic neuron. So from that standpoint, we can think of that competition as a kind of plasticity. And for those inputs that are not acquiring sufficient trophic support from their target, the neuron itself may very well die. In fact, for many parts of the developing nervous system, including the motor neurons of the spinal cord, we probably make about twice as many neurons that ultimately survive. And those that do not survive die for lack of trophic support. So there's one example in the embryonic nervous system where plasticity can lead to the death of a neuron. Now, I suspect the question really is about the more mature nervous system, and can the depression of a synapse, for example, lead not just to the loss of connections, but to the death of the neuron that grew those connections? That I seriously doubt, only because of the richness of connections among more mature neurons. So, for example, a typical neuron in the cerebral cortex is probably interconnected with at least 2,000, maybe 10,000 other neurons. So while some of its connections are likely being depressed at any given time, you can be quite confident that other connections to other neurons are being strengthened. So even if there is a kind of competition for trophic support that's happening in one set of terminals, there is similar competitive mechanisms happening at other sets of connections. And it's highly unlikely that any one neuron is going to be so deprived of trophic support from all its synaptic partners as to be lethally injured by that trophic loss. So my general sense of the question is that in the mature nervous system, no, synaptic plasticity will not lead to the death of a neuron. So we do need to wrap this up, but maybe one last question.
Gustavo that just popped in.
Okay, let's try Gustavo. Are you there, Gustavo? I think you're muted. So if you are there and could unmute yourself. Otherwise, maybe one more chat question or something someone has written in, Nick.
One question. I wonder if joking is neuroscientifically such a higher cognitive feature that is typical only for humans?
That's a wonderful question. I am inclined to suggest that the ability to joke or to jest really is a social phenomenon, and humans are not the only creatures capable of rich social experience. So it would not surprise me if we found something like a joke in a more primitive form, perhaps, in other kinds of social creatures. And this might include other kinds of primates. It might actually include some bird species, such as the corvids, that seem to have very rich social relations with conspecifics and really just very rich behaviors that are more similar to what we see in some primates. So it wouldn't surprise me if we found something like what we recognize as joke or jest. That's really a great question. I'm going to have to dig deeper into this. I'm actually scheduled to give a talk in a conference on humor, so I'm being asked to talk about the neuroscience of humor. So I will try to dig into this just a little bit, and if I can find something, I'll be sure to include it as a post in Medical Neuroscience. So thanks for that question. And I think we need to sign off, so thanks to all of you that have joined us here in the Hangout, and for those of you who've just been watching, I hope this has been a good use of your time. So thanks, everybody. I look forward to the next weeks ahead as we continue our journey in medical neuroscience. Okay, best of wishes to everybody.
Thank you.
All right, thank you all. Bye-bye.